Good afternoon, or good evening, or good morning, everybody, wherever you are in the world. Welcome to our OpenFlow webinar. It's the first one of 2022, so welcome back. I see a few familiar names in the attendee list, which is great. Today, we're going to be talking a little bit about DNA analysis by flow cytometry. And as usual, even if you never do DNA analysis, what we're going to hopefully try to give you is the principles of sample preparation, thinking about fluorochromes, thinking about how we set up the cytometer to run the samples, and also how we're going to analyze those samples as well. As usual, if you want to get in touch with us as we're going along, please use the Q&A function if you've got any specific questions, and we will try and stop and answer those as we go along. If there are any other issues, there's also the chat function that you can use as well. So who are we? Well, my name is Derek Davis. I am the Science Technology Platform Training Lead at the Francis Crick Institute here in what earlier was sunny London, a bit dark now. And I'm joined here by my colleagues across the pond. So I'll hand over to Kathy.
Thank you, Derek. So hi, everyone. Again, nice to see some familiar faces here. So I echo what Derek just mentioned. So again, I'm Kathy Daniels, and I work as the Flow Cytometry Core Facility Manager at the Whitehead Institute here in, I just had to check, not so sunny, cloudy Cambridge, Massachusetts, but it's not snowing. So happy about that. And Bree Gardner was not able to make it today. He offered his apologies, but he's happy to be joining us for our next session that we'll tell you about a little bit later. But we miss Rui today, but he's here with us in spirit. Thanks.
Excellent. Thanks, Kathy. So what we're going to do today, or this evening, this afternoon, is, as usual, I'm going to start off just giving you a little bit of theoretical background, which sort of fluorochromes we might use if we want to look at DNA analysis, how we need to think about sample preparation. And then I'm going to hand over to Kathy, who's going to run some samples on the flow cytometer. So we'll look at how we set up the machine and considerations of things like how many events we might want to acquire and so on. And then we'll look at some analysis of the data. So we're going to be using FACSDiva to do the acquisition. We're going to be running it on a FACSAria. And then we're going to do some practical analysis of the data using FlowJo. And right at the beginning of today, a big shout out to Quan Chung, who's actually provided the samples for today's demonstration. And he's in Cheeseman's lab at the Whitehead Institute. So thanks very much, Quan Chung, for that. So I suppose the first question that we have to ask with any application of flow cytometry is, why are we doing this? Why are we looking at DNA analysis in the first place? Most of you are probably in a research environment, and I'm sure you're culturing cells. So, we're all used to counting our cells, aren't we, to know that they're proliferating. And sometimes that's a laborious technique. We might be using a hemocytometer, or we might be using an automated counter. But we can do a similar sort of thing on a flow cytometer, and a flow cytometer actually gives us more information. We could potentially look at subsets of cells and see how they are proliferating. And it also gives us the opportunity to see if there are any effects of anything that we do to our cells. It might be treating them with drugs, it might be removing the serum, and so on. Flow cytometry allows us to get a little bit more information, and we're going to do this by using a fluorochrome to look at the DNA content in our cells. This is something that's actually fairly widespread, both in the research field and in the clinical arena as well. Of course, as usual, it's not the only way you can look at DNA content. You could look at chromosomes, you can look at metaphase spread cytogenetically, and there are a whole raft of image analysis techniques that allow you to measure DNA content as well. Obviously, we like to think that flow cytometry is the best way of doing it. So if we think about a cell is a cell, and if we give it some signals, that cell can do one of several things, can't it? If the conditions are right, it can divide, it can start proliferating, and that's exactly what we want during embryonic development. It's what we want whether we're regenerating or when we're wound healing. Sometimes, of course, the conditions aren't right and cells can choose to die, and they can die in many ways. They can go through a process known as apoptosis. Many of you will be familiar with that, or another process called autophagy. Again, many of you will be familiar with that. Sometimes cells just die randomly, a bit of injury to a tissue and become necrotic. Or it could neither proliferate or nor die, and a cell could enter a process of senescence. Now, all of these things are very important biological phenomenon, but we're going to be looking mainly, I suppose, at this one today. And we're mainly going to be looking at mammalian cells. Apologies to those of you who are using yeast or other organisms out there. And many of you will be familiar with this sort of cartoon. This gives you an idea of how cells replicate. So cells that are, most of the cells in our body have done their growing, and they're in what we call the G0 phase of the cell cycle, but they still retain the ability to divide. And the majority of our somatic cells will have a diploid content of DNA, 46 chromosomes worth of DNA, which we call a diploid content. And cells that are in G0 and cells that are in G1 both have a diploid content. The difference is that these fellas are sleeping, I suppose, and cells in G1 are getting ready to divide, but they have the same DNA content. When a cell makes the decision to divide, what it has to do, of course, is replicate its DNA. To be able to divide into two daughter cells, we need to double our genome. And that's what the cell does during S phase, the synthetic phase of the cell cycle. It's making DNA. And it continues to make DNA until the genome's doubled, and then the cell enters G2, has a little bit of a rest, and then moves through to mitosis. And of course, then we get division, and we get two daughter cells that now take half of that doubled amount of DNA and either go back to a resting state if your repair of your paper cut is finished, or more likely if you're using cell lines, those cells will go back into G1, ready to divide again. Cell division is one of the most, or probably the most dangerous thing a cell can do. So we know that it's very tightly controlled, particularly at these transition phases. A cell won't enter S phase unless it knows that all the machinery is there to double its DNA and enough protein is there to go into the daughter cells. A cell won't enter mitosis unless it's checked that the genome has been faithfully reproduced. And all of these things that control those transition points are proteins or enzymes. So we can use flow cytometry to look at those. And a couple of Nobel Prizes in recent years have been given for developing or understanding our developing our understanding of how cells go through the cell cycle. But we can be simpler than that, can't we? Because we can look at the amount of DNA in cells. I already said that cells in G0 and G1 have a diploid content of DNA. They have the same amount of DNA. So if we stay in them with the DNA binding dye, what we should see is a G0, G1 peak. Cells that have doubled their genome, cells that are in G2 or that are mitotic, that are still together, that haven't actually come apart, they will have twice as much DNA. They'll have a tetraploid content of DNA. And the cells that are in S phase that are synthesizing DNA will be somewhere in the middle here. And hopefully many of you are familiar with this sort of DNA histogram from the literature. And in fact, DNA analysis was one of the very first applications to which flow cytometry was put, even way back in the late 60s, early 70s. And in fact, this is where I first started. This is a BD fax analyzer, and it enabled me to produce a DNA histogram. Here's our cells in G1, S, and G2. And I'd come from a point where I was doing this DNA analysis by image analysis, and it used to take me 2 hours to construct a histogram like this. In a flow cytometer, I suddenly found that I could do the same thing in a few seconds, or at least a minute or so. Changed my life completely, which is why I'm still here. You know, flow cytometry is a great technique. But in order to make that DNA histogram, we need a fluorescent dye. as we do with any flow cytometric application, we have to make the part of the cell that we're interested in fluorescent so that we can see it and we can quantitate it. And here are a list of some, not all, of the DNA binding dyes that you might be familiar with in flow cytometry. Maybe you've used prapidium iodide or DAPI or the Herx dyes. Now, these all have different excitation and emission characteristics, and obviously that's an important consideration. But they have one thing in common, and that is that they bind in proportion to the amount of DNA that's in the cell. They bind stoichiometrically. And that's important because I want a cell that's in G2 to take up twice as much dye and therefore be twice as fluorescent as a cell that's in G1. As well as having different excitation and emission characteristics, these dyes, will also bind slightly differently. Now, we don't necessarily need to know about the binding, although in some situations it does become important, and I will touch on it a little bit later on. So dyes like prapidium iodide are intercalating dyes. They just simply sit in the double helix there. DAPI is a minor groove binder. Many of the Herx dyes, in fact, most of the Herx dyes are what we call bis intercalating dyes. They bind to AT pairs or AT-rich regions of DNA. And the reason why it's important to understand a little bit about the binding will become evident, I hope, in the next slide. So which of these dyes would you use? Well, as usual, we have to think about our cytometer. Which lasers do I have at my availability? As we know, pretty much every flow cytometer out there will have a blue 488 nanometer laser, and that's pretty good for exciting prapidium iodide, 7AD, some of those other dyes as well. Most cytometers will also have a red laser, which brings TOPRO 3 into the picture. And if you've got a violet laser, or if you're lucky enough to have a UV laser, that means Herx and DAPI are able to be used. And this is great because it gives us a choice. Sometimes we're going to be using a DNA stain in combination with a fluorescent protein or another antibody label. So the fact that we have some choice is great for us as cytometrists. Sometimes we want to think about the specificity of our dyes. As I said, some dyes don't really have any major specificity. They just happen to be the right size to intercalate into the DNA helix. But some dyes are specific. As I already mentioned, Herx binds to AT pairs. Things like chromomycin bind to GC pairs in DNA. And that was actually very important when some people still do this, use flow cytometers to analyze chromosomes. Now, I'm not going to go into that at all, but the use of two DNA dyes that bind to different parts of the DNA is very useful in chromosome cytometry. Just pop back one slide. Most of these dyes here require our cells to be fixed or permeabilized if we want to quantitate the DNA. Now, that's great if I just want to get a bit of analysis done, but sometimes I want to get those cells back. I want to sort them. I might want to sort cells that are in G2 or in S to do something with them. In which case, I'm probably not going to use a dye that needs my cells to be fixed, because if they're fixed, they're dead. And there's a smaller cohort of dyes that we can use if we want to quantitate the DNA in live cells. Originally, this dye Herx 33342 was the only dye that we had that would bind to the DNA in viable cells, which was great, but it really requires a UV laser to be optimally excited. And of course, 20, 30 years ago, we didn't have UV lasers really on our analytical cytometers, at least. But maybe 20 years ago, Biostatus developed this dye called DRAC5, which again enters live cells without the need for fixation or permeabilization, but it's excited by a blue laser and a red laser, in fact. And more recently than that, molecular probes have come out with the dicycle dyes, a whole range of different dyes with different excitation and emission characteristics. So we have a bit more choice now. And actually, all of our DNA dyes, we can use them in two ways. We've discussed at some of our other webinars the importance of using viability dyes in our flow assays. Many of our DNA binding dyes are used as viability dyes. If I've got a population of cells, some alive and some are dead. If I add a dye like propidium iodide, it'll go into the dead cells, but not the live ones. So I can separate my dead cells from my live ones. I'm not going to talk any more about that today, because this is what we're mainly using these DNA binding dyes for today, to measure and monitor the DNA in our cells. Now, if you think back to that cartoon of the cell cycle that I showed you, what you might expect to see on the cytometer is something that looks like this sort of modified rugby goal or American football goal, whichever way you want to look at it. You know, all my cells in G1 here have the same amount of DNA. They're all diploid. So in theory, they should all take up the same amount of DNA binding dye. They should all fluoresce with an equal intensity, and they should appear in a single channel in our flow cytometer. Cells that are in G2 will have twice as much DNA. They'll take up twice as much dye. They'll be twice as bright, but again, they should all appear in a single channel. And then cells that are synthesizing DNA, cells that are in S phase, will be somewhere in the middle here. But of course, we never live in the ideal world. We're in the real world. And what we see in the real world is a little bit of data spread. We can still see in this histogram here, our G1 population, our G2 population is about twice as bright, and our S phase cells in the middle. But our G1s aren't in a single channel. We see that there's a little bit of a spread. And something we talked about before is how we measure data spread. And we can do that by measuring the CV, the coefficient of variation, or the coefficient of variance, which is a statistical term. It's defined as the standard deviation divided by the mean expressed as a percentage. But in practical terms, I suppose, we want that to be as low as possible. If I look at this G1 peak, I want it to have as small a CV as possible. And that's because when it comes to the analysis, here is a bit of a gray area, isn't it? Are these cells in G1 or are they in early S? Similarly here, are these cells in late S or are they in G2? The tighter I can get my peaks, the easier it's going to be to answer that question. But we know also that we're always going to get variation. And that variation is going to come from three sources. And this is not specific to DNA analysis, but it's possibly slightly more important in DNA analysis. We're going to get some biological variation. If I take two cells that are in G1, they've got the same amount of DNA, but their DNA conformation may be slightly different. Maybe this one's a little bit tighter. This one's a little bit more open. I'm going to get a little bit more DNA into the more open one than the more closed one. So there's going to be some natural variation there. We're going to introduce in variation by what we do to our cells. I said that we're going to have to fix or permeabilize our cells to look at their DNA content. That fixation and permeabilization can introduce some variability, as can our dyes. So just as we titrate our antibodies, we also need to think about titrating our DNA binding dyes. And we can also introduce variation by our instrument setup. Again, we've seen in one of our earlier webinars the difference that we get when we press low or medium or high pressure. And we'll look at that today practically. We know these are all going to cause issues, so we're always going to try to minimize this data spread. So the first thing we can start thinking about is how we're going to prepare our cells. Remember, a flow cytometer is a single cell analysis instrument. But to stain the DNA, mostly we're going to have to fix our cells. And if we're going to fix them, how are we going to fix them? So a couple of different ways. Firstly, we can use alcohol. By alcohol, I normally mean ethanol, occasionally methanol. And alcohol does two things. It dehydrates, it extracts water from the cells and from proteins. So what that means is that it coagulates proteins. Now, this is great for DNA analysis because normally our DNA is surrounded by all these sort of globular proteins. And if I shrink them by fixing an ethanol, it means that my DNA dye can get into the DNA fairly freely and it minimizes the variation. Alcohol also permeabilizes because it dissolves lipids, and that's great because, again, it means my dye can get into the cell very easily. And if I'm just looking at DNA analysis, I would probably use ethanol because it gives me the lowest CV, the lowest spread of data. In practical terms as well, if I fix my cells in ethanol, I can store them for quite a long time before I need to stain them. And that's great, obviously, because it means if you're doing a longitudinal experiment, you can wait till the end of the experiment to do your staining. You don't have to do it as you're going along. And fixed samples are stable in alcohol for months. In fact, next week, I'm going into the lab to fix a whole pile of cells that I will use in demos throughout the year. Now, there they are, that stable. Pros, there are always cons, aren't there? And the cons of alcohol is that it's not going to be that compatible with fluorescent proteins. If you have GFP in your cell, for example, and you fix in ethanol, you're generally going to lose that GFP fluorescence. Same with some of our protein-based fluorochromes. We have to be a little bit careful if we're ethanol fixing. And all fixation will increase autofluorescence. Doesn't matter quite so much with DNA analysis, because as we'll see, our stained cells are way, way, way brighter than our unstained cells. But if I'm doing surface staining at the same time, that can have an effect. If I don't want to fix my cells in ethanol, I can fix them in an aldehyde. Generally, formaldehyde is the fixative of choice, and formaldehyde is a cross-linking fixative. So all those proteins that are around DNA get locked in place because they're cross-linked. Now that means that it's slightly more difficult for my DNA binding guide to get into the cell, and that leads to a higher CV. But aldehyde fixed cells are more compatible with fluorescent proteins and other fluorochromes. So as usual, it's going to depend on the question that you're asking. If you're just looking at DNA analysis, I would normally recommend alcohol. If we've got fluorescent proteins in there, I would probably recommend formaldehyde. We can't store aldehyde fixed cells for quite as long as we can alcohol fixed cells. In fact, what I normally do is I fix my cells in formaldehyde for 20, 30 minutes, wash the formaldehyde off, and then store the cells in PBS with a little bit of azide. So it means that they're stable for a couple of weeks or so, but I certainly can't keep them for quite as long as with alcohol fixed cells. I said alcohol is a good fixation, it's a good permeabilizing reagent. Formaldehyde is a reasonable fixative, but it's a fairly poor permeabilizing reagent. So generally, we're going to use a permeabilization step. And those of you who are used to doing intracellular staining for flow cytometry will probably already be familiar with the fix and perm kits that you can get. We fix it formaldehyde, and then we put a detergent on to help permeabilize them. And again, we're going to get some background autofluorescence. Actually, we don't have to fix at all. We could just use a detergent. We could just use a permeabilizing reagent. Many of you again are familiar with Triton X100 that you might be putting onto your cells before you do a Western to pop them and release the proteins. We can do that in DNA analysis. We can put our cells in fairly high concentration of detergent, which basically removes all the cytoplasm and leaves us just with nuclei. Again, that helps with the CV. But you generally have to run them fairly quickly. I mean, you can fix them and run them later, but normally I would run them straight away. I'll just put a couple of references or a couple of resources in here as well. This is it's quite an old paper from current protocols in cytometry from Zizek Darzenkiewicz and Gloria Juan. But a lot of the protocols have not really changed for many years. So this is a great resource if you can get hold of it. And this URL here points to a talk I gave at Flowtex last year, talking about fixation and permeabilization. So Kathy has appeared as if there's some sort of question that I need to answer.
Yeah, so we actually have two great questions that came through. Actually, three. There's one I think that you're going to cover in a little bit, so we'll hold off. But one question from Priscilla that came through is, what is the difference between formaldehyde and paraformaldehyde in terms of flow cytometry?
Well, that is a great question, and it's one of my biggest bugbears, okay, because you will read in the literature and people will always say they fix cells in paraformaldehyde. Okay, so this is not true. If you look at a pot of paraformaldehyde, it's a white powder. If you put it in a PBS, it remains a white powder. It's insoluble. If we manage to get that into solution by heating it or by treating it with sodium hydroxide, paraformaldehyde is polymerized formaldehyde, And what the heating does is it chops those bonds, and we end up with formaldehyde. And formaldehyde is what we fix in. And actually, I use a ready-made formaldehyde solution from Polysciences, although a lot of other companies do that as well, because actually getting paraformaldehyde in solution is quite a messy and prolonged business. Okay. So yeah, it's a semantic thing maybe, but it's my hobby horse. Now, you fix things in formaldehyde, not paraformaldehyde.
Yeah, I should say those are the facts, but that brings us to a conversation that's happening on
another
to-do list right
conversation.
now. Yeah, so let's steer clear of that and go to the next question. So Brian Hanley is asking, does the amount of time spent in formaldehyde fixative affect the autofluorescence, and also does this saturate over time?
Absolutely. Well, the first part of that, yes, is definitely true. Yeah, one of the reasons why I do the fixation and then wash it off, because the longer you leave things in formaldehyde, the more the autofluorescence increases. I've never actually done the experiment to see where it saturates because I'm too scared to do that. But you know, it's a good habit to get into. It doesn't matter how long you fix for, really. I mean, anywhere between 15 and 60 minutes or something like that. But keep that consistent. Keep that fixation time consistent, and remember to wash it off at the end of it, because it will increase the autofluorescence. If you're just looking at DNA, that probably doesn't matter too much. But if you're looking at weakly expressed markers as well, it can make a big difference.
Agreed 100%. And the third question that that came up, I think you're going to be covering it in a little bit, but just to give you a heads up, Masume is asking if cell count is important in spreading. So there's some curiosity about that, but that's it for questions now.
Okay, yeah, we'll probably cover that when we get to the practical thing. We'll talk about cell numbers. Yeah. And Aaron's just saying that link is unavailable. I'll check that as we go along, and I'll put it into the chat.
Sounds good.
Okay, so one of the reasons I like DNA analysis is it's quick and it's easy. And if you're using mammalian cells, I can give you a protocol that will pretty much work for most mammalian cells. But as usual, if you come to a new lab, you're a new PhD student, you're given a protocol, you run through it, hey, you get a result. What you should, of course, be doing is looking at those stages of the protocol and thinking, well, why am I doing that? What's the purpose of that? And this sort of 6-step protocol here for DNA analysis, if we stop and think about it, actually, there's some things in the background that are important to think about. So I guess I've glibly put here, harvest cells as normal or as usual. Of course, that depends completely on the cell type you're using or the type of tissue you're using. If you're using suspension cells or peripheral blood, your harvesting is going to be very different to if you're using adherent cells, or if you're taking cells out of paraffin wax, or if you're taking cells out of a tissue in an animal. This is your realm, though. Generally, our users are best placed to do their sample preparation. And sometimes that can take quite a lot of time, because remember, we need a single cell suspension for flow cytometry. Once you've harvested them, I say fix in cold 70% ethanol. So there's several questions you might ask there. What does cold mean? Well, cold can either be fridge cold, so 4 degrees, or freezer cold, minus 20. And it doesn't make a big difference which one you use, but I normally use 4 degrees because I've got more fridges than I have, more fridge space than I have freezer space. And the reason it's cold is because the process of fixation is exothermic. We don't want our cells to boil or to change their morphology or their makeup during the process of fixation. So we use a cold fixative. 70% ethanol. How do you make up 70% ethanol? Well, it's important to make it up in distilled water. We take a solution of 100% ethanol, absolute ethanol, and dilute it to 70% in distilled or deionized water, not PBS. Okay, if we make it up in PBS, what will happen when we add it to our cells is we'll get phosphates precipitating. We'll get precipitate in there, which is not good. You might think, well, I've got 100% ethanol, haven't got much time. 100%'s got to be better than 70, right? It's a bigger number. Also not true. That little bit of water in there actually helps the ethanol get to all parts of the cell, the hydrophobic and the hydrophilic parts of the cell. So it doesn't have to be 70, anywhere between 50 and 80 actually works quite well, but traditionally we use 70% ethanol. And another thing that sort of gets hidden in this protocol is how we should fix. What I normally do is I pellet my cells. I take my pellet, I put the cells on a vortex, and I add my fixative as quickly as I can. The rationale there is that we're staining all the cells at the same time, so we should get even fixation, and that's going to help us with that data spread as well. If all cells are evenly fixed, we should get a lower CV. We should keep our cells in these fixative for at least 30 minutes. It does take, alcohol is a small molecule, but it does take a little bit of time to get through the cytoplasm, through the nuclear membrane and so on. So 30 minutes is sort of a minimum to do that. It's one of the reasons why the permeabilization method is sometimes quite useful. Sometimes you don't want to wait 30 minutes. You want to get a quick look-see about how your cells are proliferating. And using that detergent method is sometimes quite useful because you can put them in Triton, stain them and run them almost immediately. But if you're fixing in ethanol, try and leave them for at least 30 minutes before you do the rest of the staining. And the next thing is to wash your cells off, remove that alcohol. To be honest, you could stain your cells in the alcoholic solution and run them through the cytometer, but you'd have problems because your sample fluid and your sheath fluid will have different viscosities and different refractive indices. And that'll lead to problems in the laminar flow, and it'll lead to problems in light scatter. So we should always try to make sure that our cells, once they're stained, are in a fairly similar type of fluid to your sheath fluid. Generally, we're also going to add RNAs. Most of certainly our intercalating DNA dyes will bind to many helical structures, including double-stranded RNA. So it's not always needed, but I always put that in. It's a bit of belt and braces. Put a bit of RNAs in to remove any double-stranded RNA. However, so some of those things, PI, pyridine iodide, also binds to keratin. So with keratinocytes, you have to be a little bit careful that you may get a little bit of non-specific binding. Another reasons why sometimes using a permeabilization technique to release nuclei only can be useful. And then we add our DNA binding dye. And for mammalian cells, pyridine iodide, 50 micrograms per mil is what I would normally recommend. But if you're using plants generally have a bigger genome size than mammalian cells. You might need to go higher. If you're using yeast, which have a much smaller genome, we could go much lower. So it does depend on the type of cells that you're using. Importantly, we shouldn't wash this dye off. Those dyes are not binding covalently to DNA. And if we wash them after the staining, what will happen is we'll start getting equilibration of the dye. It will start leaving the DNA. So again, that will contribute to the data spread. So we're going to run our samples today in the staining solution. So this is just showing you that you can titrate our DNA binding dyes in the same way that we titrate antibodies. So what we're looking at here in light blue is the staining intensity. So as we increase the concentration of the dye, what we see is an increase in the intensity of the staining, because obviously more dye, we're getting more dye into the DNA, our signal is increasing until we get to a sort of plateau phase when we're saturating the DNA. But we're also looking in dark blue at the CV, the spread of the G1 peak, and you can see at low concentrations that spread is quite big, so very high CVs, but that comes down as we start to saturate our DNA. So normally around about here, 50 micrograms per mil is what we use for human cells. That gives us a bit of leeway if our cells are maybe slightly more or less concentrated. So now we've thought about sample preparation, a bit about staining, we have to think about the cytometer. So again, we've talked in previous webinars about the importance of QCing our flow cytometer. For DNA analysis, this is crucial. We really need to make sure our cytometer is working optimally, that our lasers are properly aligned. And we do this using all sorts of beads, whether they're 6-peak or 8-peak or BDCS and T beads. Because if we've got a misaligned laser, that shows up very quickly. We get a much broader CV in our beads. And when I'm doing DNA analysis, I always actually run a bead QC check before I start my experiments, because I want to know that the cytometer is performing correctly. As long as it is, okay, I go ahead and run my samples. Again, something we've touched on before, and we'll see in a moment when we go to Kathy at the cytometer. You're aware that in all of our flow cytometers, we can change the pressure that we apply to our sample, low, medium, or high, which affects the speed that we put fluid through the cytometer. But at that high flow rate, we get more variation about where the cells go through the laser beam. So with DNA analysis, we should always be running our samples on low. Occasionally, you'll see in the literature, don't run your DNA samples at more than 500 per second. That's not true. I can easily run 5,000 per second as long as I keep it on low. That's the important thing. Because this cartoon shows why. If I'm on low sample pressure, my cells are being hydrodynamically focused, squeezed into quite a narrow core that goes through the same part of the laser beam each time. So I get a reasonably low CV. If I take exactly the same sample and change it to high flow rate, I get a wider core. So now there's more variation about where the cells go through the laser beam, and there's more chance of more than one cell being in the beam at once. So I've got a much worse DNA profile. It's the same sample, just because of what I've done on the flow cytometer. The other thing we need to think about, again, in all experiments, is removing doublets. And something we've shown before, what happens as a cell goes through a laser beam, as it enters the beam, we start seeing a pulse being produced. When it's in the beam, that pulse increases. And then as it exits the beam, that pulse decays. And we can measure three things: the area of that pulse, the height of that pulse, and the width of that pulse. Again, we'll look at this in more detail in a moment. But if we plot the pulse height against pulse area, and normally we do this for forward scatter. But if I've got a DNA dye in there, I normally use my DNA dye to remove doublets. So if I plot height and area of my DNA dye, I should see my single cells fall along the diagonal, because whether a cell is in G1 or S or G2, the area and the height are in proportion. If I have two cells coming through very close together, I get this sort of camel hump effect. And that means that the cells or the events will fall off that diagonal. So I can exclude those, because remember, we're a single cell technology. We want to make sure, as far as possible, we're just looking at single cells. We'll talk more about this maybe a little bit later on. It's not 100% perfect. And this is what this looks like in real life, if you like. So having thought about our cells, our dyes, our sample prep, and our cytometer, hopefully we're going to get something that looks like this, our DNA histogram. Cells that are in G1, certain amount of DNA. Cells that are in S that are synthesizing DNA. Cells that are in G2 or mitosis that have already doubled the amount of DNA. Importantly here, you'll notice that we're running this on a linear scale. When we've done all of our webinars before, when we've been looking at immunophenotyping, we've used a log scale for our fluorescence, because I'm looking at CD4. My CD4 positive cells may be 100 or 200 times brighter than my negatives. I'm going to use a log scale to be able to display those two together. With DNA content, I'm really looking at just a doubling of fluorescence from G1 to G2. So I don't need to use a log scale for that. I'm going to use a linear scale. It's going to spread it out a lot more. Not only is it going to be aesthetically more pleasing, it's going to make it easier for us to do the analysis. Okay, so that's enough background. That's enough of me whittering on. Let's see some of this in action. So, as we've got the right, we've got the link up here. Please ask questions as we go along. I'm going to stop my share so that I can let Kathy do that.
Sounds good. Thank you so much, Derek. Let me make sure I'm sharing the right screen. And I can see I'm on two different computers today, so I can see it's sharing, which is great. And now we're ready to, perfect. Thank you. So now we're ready to go ahead and look at the practical run of DNA analysis by flow cytometry. So again, I just want to thank Hwan Chong from Ian Cheeseman's lab here at the Whitehead, who is kind enough to provide these samples for us today. You see here that we have a new experiment set up in DIVA. Now, because I'm working on a dual monitor screen right now, all we're seeing is the global worksheet, the inspector, and the cytometer window. But rest assured, I have the browser with the new experiment and our acquisition dashboard and all of that on our other screen. We are working on a BD Fax ARIA today for this data acquisition. Okay, so what you'll notice first in the cytometer window is that I've already selected the detectors that we need for our experiment. If you've joined prior sessions, you know that we can add on or remove detectors appropriately based on what we need for our experiment. So we're looking at forward scatter and side scatter. And then on this instrument, the optimal detector for PI is PE Texas Red as a label, but it's off the yellow-green laser, and it is a 610-20 filter, so it's taking light from 600 nanometers to 620 nanometers, okay? We also have a detector off of the blue laser line, which we're gonna use to show that PI is excited off of both of the blue and the yellow-green lasers, okay? Now, when you first come in, what's gonna happen is your forward scatter and side scatter is going to be in linear, and it's typically always, at the very least, in area. As Derek just described, we want to make sure that we can pull out single cells versus aggregates when we're doing all of our analysis. So we're going to enable height and width for forward and side scatter, okay? Now, the next thing that Derek mentioned that we're going to make sure that we enable for today is looking at our detectors, not in log, but in linear. So here in Diva, all we have to do is uncheck log, and then that's going to give us a linear display in the software. Now, we also want to make sure that we can look at height versus area for our fluorescent output as a double doublet exclusion. So what we're going to do is we're going to check off to have height, area, and width in linear for essentially every detector that we're using for this experiment, okay? Now, that being said, let me go ahead and set up our first plot that we're going to use today. So I'm going to select right here, and that's our dot plot. I'm going to click and drag. And our first plot is going to be forward scatter versus side scatter, because when I load the cells, we're going to first try and get them on scale by scatter. Okay. Let me just modify this so that hopefully everyone can see it a bit better. So in the inspector, under the labels tab, you have the option to make it a little bit bigger, and hopefully everyone can see this. So we're going to make everything a little bit easier to see. Okay. Now what I'm going to do is I'm actually going to take the sample and I'm going to load the first sample up so that we can actually start using the events as they're coming through to set our gates. If you have a lot of experience in flow cytometry, it might be easier to set a template up and understand where your cells are probably going to fall. But if you're new, especially to this type of analysis, load your sample and start setting up your gates based on the events that are coming through. Okay, so the first sample that we're running, as you see my back as I'm going ahead and vortexing, is a methanol fixation sample that we have stained with PI. Okay, so this is our control sample. It should have a normal distribution. I'm keeping this at the lowest flow rate for the ARIA, which is 1. And I'm going to go ahead and load this sample onto the ARIA. Okay. Now I have the name as methanol host, but that's not the case. It's methanol fixed PI. So I'm going to change that in the browser, and you can see that that's changed right here. All right. So if I bring over the acquisition dashboard, what we can see is that we have events coming through at about 200 events per second right now, but I don't see much of anything right here in the scatter plot. So we've went over this before, but a little trick that I like to teach people is if you create a gate and bring it against the axis, and then you click on the plot itself, right click and show population hierarchy, you get an idea of where your cells are. So 80% of your events are falling within this gate. That tells me the side or the forward scatter is too high at a very minimum, and likely the side scatter as well. Okay, so what I'm going to do is I'm going to go ahead and I'm going to reduce the forward scatter. Let me try about 300. Then on the acquisition dashboard, I'm going to restart the analysis. Okay, so we see that they're still off scale, so I'm going to bring that down, maybe do about 150. And as I do that, we can start seeing that we have a higher population of cells that are up here. 96%. So now I know I need to reduce my side scatter. So let me bring that down to about 260. I'm going to restart so that we refresh and only see the most current events. And now we see if we adjust this gate, that's a majority of our cells, okay? You also can tell on the threshold that it's set right now off of forward scatter and 5,000. So we are able to see anything right about here or below. Okay, we're going to maybe potentially get into thresholding a little bit later if we have some time. But for now, we're able to see a majority of our cells, and this is exactly what we want. Okay, so there are some cells off scale right here. If you wanted to, you could make a slight adjustment and bring that down to 140, maybe bring the side scatter down to 250 and restart. But I think that this is pretty good. And likely when we do the rest of our gating, we're going to find that most of those are aggregates. So then I'm going to go ahead and create my first gate, which is a polygon. And I'm going to make a pretty broad gate to start off with. And I'm going to include these cells that are higher in scatter. Okay. I've shared this trick before, but if anyone's interested, if you hit the shift key on your keyboard and select a node, if you move slowly, you could make changes to the whole entire gate and you don't have to drag in each individual node. Okay. It's just a nice little trick I like sharing because I didn't know about that one for a while. I'm going to go ahead and I'm going to rename that scatter. And then I'm going to highlight that gate, right click and drill down, move this to the side, and now it's only showing me what's in that scatter gate. From there, we're going to do our first singlet gate, which is going to be forward scatter height versus forward scatter area. Bring this population hierarchy down. And I'm going to do my second gate, which is going to be a singlet gate. I highlighted the polygon option, went all the way up and over. And this gate right here is going to be my forward scatter singlet gate. Okay. Next, we want to make sure that we're really, really, really only looking at single cells. So I'm going to highlight this gate, right click and drill down. And then we're going to take a look at our PE height and our PE area. Okay, PE Texas red, rather, I should say. Now, we're not seeing anything right now. You might say, oh, is your laser down? Is something going on with the instrument? Nope, that's not the case. What we actually haven't done yet is we haven't adjusted our voltage for the fluorescent detector that we're using to pull out the PI. So I'm going to go ahead and generate a histogram. And I'm going to change the axis to PE Texas red height. Now, if I use that same trick from before, and I do an interval gate, and I bring it over here to the edge, that's 99.8% of my event. So I know that that signal is too high. What I'm going to do is I'm going to bring that down from 550. Let me try 400. And I'm going to refresh. And now we're starting to see our profile. Now, you might be curious and want to know where to set your G1 peak. Typically, you set that right around 50 on a BD instrument, where you, and that's the lower or the first quarter of the plot. This gives you room to see the distribution of your G1, S phase, and G2, and it also allows you some room to see any potential aneuploid cells. Okay, so let me delete that gate, and I'm going to modify this voltage and bring it down a little bit more. And you want to be mindful that it's actually pretty sensitive. So if I went down to 300 and started over, that makes a very big difference. Okay, so let's try about 330. Restart and see what that looks like. It's a little low still. So I'm going to go ahead and bring that up to 350. That looks pretty good. I'm a little anal. So I'm going to go up and maybe bring that to 353. And that looks great. Okay. What I'm also going to do just quickly to show you is I'm going to show you how to look at that off of the blue laser as well. So if you didn't have an instrument that had a yellow-green laser and you were looking off the blue, you would follow those same principles. PerCP Sci-Fi 5, that's the detector that we have that is able to pull out the PI signal on this instrument. And I'm going to bring this signal down. I'll try 350. And it's actually pretty similar as far as the voltages go. Okay. That's not always the case, but for this example, that was in fact the case. Okay. So that's just to show you that you can pull out the PI off of both lasers. Okay. Now, I'll probably go forward next and change this color because I recognize the neon green is not the easiest for everyone to see. So we have our scatter gate, our first singlet gate, and then here we have our second singlet gate. Now, if I adjust this forward scatter singlet gate to include those aggregates, you see that same exact pattern that Derek was talking about, even though he plots the data with PE Texas red on the Y and PE Texas red height on the X, right? So area on the Y, height on the X. As long as you're gating the correct population, don't worry about it, right? You want to make sure that you're including the PI positive, any potential aneuploid cells, and that you're cutting out the aggregates, right? So let's change that color again, change it to orange. For any potential New Yorkers that are here, these are very Mets colors. I don't follow baseball myself, though. All right, so now we just showed you this, that you could see what that pattern looks like by PI, but I'm gonna bring this gate in so that we're only looking at the single cells, okay? So that's here. I'm gonna rename this gate and call it PI singlets. And then what I'm gonna do is I'm just gonna go ahead and change this back to the preferred detector, which is off of the yellow-green laser line for this experiment, okay? Now, Derek mentioned that as we change the flow rates, what can happen is the CV of the data can expand, right? We can see a broader CV, and we don't want that because we're trying to get nice tight peaks to be able to identify these populations. So if I change the flow rate, I'm just going to check my sample volume. We have a nice amount left. I'm going to go up to one of the higher flow rates. And I'm also going to just show in this histogram by right-clicking on the plot, show population. We're just going to look at singlets. We're going to see what it can potentially look like. Now, there's not too much of a difference. Sometimes certain instruments are a little bit better than others, but what can wind up happening is as you increase that flow rate, as you're widening the core stream, the CVs can potentially change, right? I'm going to bring it down to a lower CV, or I'm sorry, a lower flow rate rather. And then what we're going to do is we're going to be able to go ahead and start recording. Now these samples have been sitting a little bit, so what I'm going to do first is I'm going to unload it and vortex it and put it back on and run it at that lower flow rate, and then we'll be good to go. Now, for today, we do have a variety of samples to run. So this is our optimized ideal sample with the preferred fixation of our alcohol fixation. As I'm reloading the sample, Derek, are there any questions that have come through up until this point?
Sorry, I have to unmute before I can say that. Not,
Okay.
not at the moment.
Okay.
I suppose the other thing, Kathy, that we talked about before was the plot of forward scatter against the PI signal.
Oh, yes, good point.
Run.
Very good point. So this is another plot that we can look at. So if we look at a dot plot, let me actually drill down from the singlets rather to get a better visualization with the modified fonts. What we could do is we can actually take a look at our forward scatter on the X, and we can look at our PI signal on the Y, and we see nice, clean, straight populations indicating the instrument's performing as expected, right? So we see these nice, very clear, very distinct populations, and we don't see any curving of the data or anything like that. So that's just what we want to see. And this is a nice plot that we can use to QC our instrument and make sure.
because we're looking at sort of high resolution data, we're only looking at a doubling of fluorescence. We want to put as many things up on the screen that will help us be reassured that the machine is working okay. So although we only have one fluorochrome in here, we've got 5 plots up when we're running our samples.
Exactly. And now that we're ready to go, a suggestion that I would typically have is to make sure that you're recording the appropriate number of events. So what we're going to start recording is 10,000 of our single cells. So I set that up in the acquisition dashboard. If I move this over here, PI singlets, 10,000. Make sure you never use the storage gate, okay? If you record the storage gate, everything else that falls outside of that gate is going to wind up not being recorded, okay? Now, another thing that we can show you as this sample is being recorded is how to look at statistics in DIVA. So if I want to take a look at the CV of this population right here, right, this G1 population, I can go ahead and I can look at this specific population. Let's call it G1 to make it easier for ourselves. And I can right click on any plot and create a statistics view. When we do that, it comes up with a random set of statistics based on what it thinks you want. That does not mean that that's what you need to look at. We can modify that. So if we right click on this statistical view and edit the statistics, what I tend to do is I uncheck all of this from the header. If you want to look at tube name, that's perfectly fine for your reports, but you don't typically need all of that information. Then from populations, what I care about is I care about G1 because I want to see the CV. So I'm only going to look at G1. Personally, I don't really, I'm not interested at this point in the percent or the event number. I really want to focus on CV. So what I can then do is I can then come to the statistics tab and I could look at that robust coefficient of variation and hit OK. And then you see that the CV of that population is 4.8, right? So as you're going along and looking at your data, if you're curious about the CV and want to see the tightness of the peaks, you can go ahead and look at that. That's also something that we're going to be taking a look at when we go and analyze our data in our analysis software when we're modeling the data. But this is just a good way for you to get an idea and see what it looks like. You could also look at percent frequent or the frequencies and the frequencies of the populations. You could look at G1, G2, and so on and so forth. Even though for traditional cell cycle like this, the preferred approach in my mind is the mathematical modeling because it helps to better fit the data. And we're going to go over that in a little bit. And I see Derek, so I will see.
I was just going to make a comment because we had that sort of unanswered question I think that we had in there about the number of cells affecting the spread. That's I suppose the same as any experiment in that we have to have we really want to get a certain number of events in a particular region before we're confident that that spreading is real. So we'd always want to acquire at least 500 to 1,000 cells in that G1 peak, which is why we normally would run 10 or 20,000 total events. That would give us enough in there to be able to get that. The other thing is managing your own expectations about what that CV should be, because it does depend on the cell type. If you've got resting lymphocytes, if we just take some blood out of our arms and do some Ficol and stain the lymphocytes, I would expect that CV to be quite low, maybe 1.5%, because all those lymphocytes are in the same state. They're generally all in G0, unless you've all got COVID, which may say might not be. But if you've got a cell lines, they're constantly growing, they're constantly moving. So there's more variation about how the conformation of the DNA is in G1. So I can't remember what cell line we said this one was, but I do a lot of work with Jurkats or HL60 cells. I expect that to be about 4 or 5. If I use something like HeLa cells, I'd expect it to be maybe 6 or 7, a bit broader. So it will differ between cell lines. So don't panic if your CV is different to what we show you here.
Yes, absolutely. And then another point about cell number too is that we didn't touch on yet, I believe, is at the staining step, you want to be very careful that you're staying consistent across the samples, right? So oftentimes people will come and say, all right, this, I see that the signal is much different. And I say, okay, did you count your cells before you put them in? And did you really have a good understanding and standardize it across all tubes? And they don't often when they have that issue. So keep in mind that at the staining step, cell number is very important as well to keep things consistent across your experiment.
Yeah, and that can be very important if you're looking for changes in DNA content. If you're treating your cells with something that maybe blocks them in early S or middle of S, you want to know where the G1 peak is. So keeping your cell concentration consistent will help you with that.
Yes, absolutely.
And another thing that you can occasionally do, you will occasionally see people doing, is adding in a standard to their analysis. So years ago, we used to use things like chicken red blood cells or trout cells, which are nucleated. And they have a known amount of DNA relative to the human amount of DNA. So if you sort of always put those in the same place, then you can compare where your peaks are.
Yes, absolutely. Absolutely. Okay, so our next sample that we're actually going to run today is going to be a sample there. They solubilized PFA, right? And then it turned into a formaldehyde fixation. And that is not the preferred method, but we're going to see what it looks like. Okay, let me just give it another quick vortex before I throw it on to make sure the cells didn't settle all that much. Throw that back on. And I'm keeping it at the lower flow rate. And let's see what this looks like. And it's important to note, I should say that Kwanchung did prep these samples for us yesterday. So we have run this before, and this is the about second time that we're running this. But it goes to show that you can actually do the sample prep, and while it's great to look at it right away, we're still able to look at this the next day, which is a nice note. Okay. So I'm going to go ahead and record this sample. And as we're recording, I want you to take a look at a couple of things. One is that the signal has shifted down a pretty significant amount. So we see that PI has actually drifted lower. And then what we also see is that there's a difference here in the scatter. So based on how you fix your cells, it can have an impact on the scatter profile of the cells. So if I have a really tight gate here on where the cells were with PFA, and then we go here to methanol, you can see that there is a change in the cells by scatter and where they appear. Okay, so I'm going to delete that. That was just showing you a reference point with that gate. But you can see that this profile looks cleaner compared to this, where we don't see as much staining with the PI and it doesn't pull out as nicely as we'd expect. Okay. Now, moving on from this, because we're running a little bit tight on time, I'm going to quickly show two different treatments that Kwanchung applied to his cells. And one of them, the first one that we're going to run, actually allowed for a rest in the M phase of cell cycle. So let me go ahead and vortex this. We're going to go ahead and load it up. Now, these two samples that we're about to run that have the different treatments are also methanol fixed. Okay, so we had one example of PFA, and now we're moving forward to, or back to methanol, I should say. Okay, so this is going to be methanol and then mitotic enrichment. I'll go ahead and load that up. And let's see what this looks like. So in order to do this enrichment, Kwanchung added 10 micromolar of STLC. It's S-trityl L-cysteine, I believe. And what happens is it pushes the spindles apart and prevents the mitotic spindles from forming. And it's a motor protein inhibitor, okay? So then what we see, if we bring our gates back up, is that we see this very clean population that pretty much is in our G2M, where we expect our G2M. So I'm going to go ahead and record those. or record this sample. But by applying this treatment and having the control that we ran first, which is our nice, clean, as expected, normal distribution of cell cycle, we can see very clearly that this mitotic enrichment worked very, very well with the 10 micromolar of the CTLC. All right, so this is just what we'd expect. Now I'm gonna cut the recording short so that we have time to do the analysis, right? But I have previously recorded these samples a little bit earlier today. So at the time of analysis, we will have 10,000 to work from. Okay, I'm gonna go ahead and stop this recording, acquisition. And then we're gonna go through to our last sample, which is gonna be a part of our hands-on run. And we're gonna look at our S-phase enrichment. Okay, take this off, vortex the sample and throw it on. Remember, if anyone has any questions, feel free to use the Q&A section. All right. Rename this sample. Okay, and load up. Now, for this enrichment, there was a 2.5 millimolar thymidine treatment for 20 hours, and it interferes with the nucleoside metabolism and it blocks the replication of cells. So that was the treatment that he carried out for this specific treatment. And you can see with this treatment, there's very, very clearly a change in the scatter profile in these cells. So that's something you want to also be out on the lookout for as you carry out any type of treatment to the cells, because you're going to have to modify your scatter gating, your singlet gating to make sure that you're actually seeing the populations you expect and recording the populations that you expect. And sometimes these treatments are going to be a little bit difficult, and you don't see that really clean pattern. And that's why having the controls in place are going to be really important to make sure that we're confident about our analysis. And I see Derek, so
yeah, while we're recording that one, Kathy, yeah, there's a question that's come through here. It's in which cases would synchronizing cells be important to the analysis?
Okay, that one, I actually might pass that one along to you since you have a little bit more experience, Derek.
Yeah, I can do that. Well, as Kathy said, and as we ran here, first of all, we're always going to run an untreated, hopefully exponentially growing sample, which will give us the standard cell cycle for that particular population. Often people will use DNA analysis to check the synchronization of their cells. So as we've done here, they've synchronized, or Wencheng has synchronized the cells at the beginning of S. What they would then do is release those cells so that they start growing again and maybe see how long they take to go through certain phases of the cell cycle. So obviously this first experiment would be a check to see where the cells are blocked or how well they've blocked. And then the rest of the experiment would be following the cell cycle after the release of that block. Similarly with the mitotic block there, that's a reversible block. So it's a fantastic block. I'm not sure I've seen anything really as good as that. Virtually everything is stuck in G2, which is great because now remove the blocking agent from there and those cells will start to grow. And then we can compare samples at different time points to both our exponentially growing control and our blocked control. So it's all about controls really, isn't it? As all flow cytometry experiments are.
Yes. Okay. And with that, we've now run all of our samples for acquisition and for the hands-on portion of this. So I'm going to step away from the instrument for a couple of seconds, and I'm going to go to a third computer for our analysis portion of that. But let me go ahead and stop my screen share.
I'll just fill in the time by doing some song and dance. And actually, I've got a couple of slides we can put in.
Perfect. Thanks, Derek.
So I'll share my screen again because we wanted to also show you how we analyze the data. Now we could do this in Diva, but the offline software, so we're going to use Flojo to do this, is actually a little bit better because what we can do in any of your analysis packages, what we want to do is we want to get some numbers, don't we? I mean, you could look at those plots and you can see that there's a difference between the control and the sample that's been blocked in S phase and the sample that's been blocked in mitosis. But we also want to get numbers. We always want to get the picture and we want to get some numbers out of it. So we could analyze our data like this by just simply setting, as Kathy did in the analysis, in the running there, simply by setting a region around our, what we consider to be our G1, our S and our G2 population. But we've got a problem, haven't we? Because we're sort of guessing, aren't we, that this is our G1, here it starts, it goes up, it comes down, and maybe it hits the axis about here. But because it's not all in one channel, because there's a little bit of data spread, we've got that gray area. We don't know where G1 really ends and S begins. Similarly, up this end of the plot as well. So although we can get numbers by drawing these markers, maybe they're not as accurate as they could be. Now, actually, we realized that way back in the early days of flow cytometry. So what's happened over the years is that very many people have produced deconvolution programs, mathematical algorithms that attempt to deconvolve that histogram. And they're based on the CV of the peaks. They're based on the difference between the G1 and the G2 population. And you can see, and we'll see this live in a second, that the software has produced a G1, an S phase, and a G2 population. And it's given us some numbers. And you'll notice those numbers are different, aren't they? Setting things by eye, we've underestimated S by quite a lot, and we've overestimated G2. Now, it may not matter which one of those you use. It may depend on, sometimes with DNA analysis, it really is very qualitative. Have I blocked my cells and which phase they're in? But sometimes we do want to get those numbers. So that's what we'll do now. I'll hand it back to Kathy. I'll stop the share that screen.
Perfect. Thank you.
Analysis. Okay, there's a question actually while you're getting to there. How do we decide when we use a log scale and when we should avoid using the log scale? So sure. As we said at the beginning, here we're looking at a very small difference between G1 and G2. It's just a doubling of fluorescence. In biological terms, that's quite a small difference. And in most of our experiments, we've got cells that are moving from a diploid through S to a tetraploid, then dividing and going back to diploid. But there are some situations when we get what we call polyploidy, particularly in megakaryocytes, platelet precursors. They actually can double their DNA quite a lot, so that you've got cells that have a diploid content, a tetraploid content, octaploid, 16 times 32, and so on. We couldn't use a linear scale in that case. We would have to use a log scale. So as always, it's up to us which one we use, and we're going to use the one that best fits the biology. So if I'm just looking at a normal cell cycle, I would use a linear scale. If I'm expecting a big range, and sometimes if you're doing plant biology, this is also the case, I might use a log scale. So you could use either.
Yes, great point. I think I remember, if I remember correctly, it's been a little bit, but doing hepatocytes a couple of years ago and seeing a similar pattern there as well, where we had to look at it in a log scale.
You get a lot of octaploidy in hepatocytes, generally not more than that. So you can probably get away with a linear scale. If it goes more than that, I would go with log.
Yes. Okay. So at this point, we're good to go and look at our analysis in FlowJo. Okay, so what we're going to first start off with is recreating our gating strategy as we did in DIVA. So we've already had sessions on DNA analysis using FlowJo. So we're not going to go over too much of the basics, but I'm going to be relatively quick here and just open up our first methanol fixed PI sample. And I'm going to create an elliptical gate around a majority of our cells. Let's scatter. Okay. And then I'm going to double click there, and we're going to do our forward scatter height versus forward scatter area and a polygon gate to look at our single cells. Since I'm going a little bit quick, you could always slow down the speed that you watch this at if you'd prefer. Okay. And then from there, what we're going to do is we're going to look at our PE Texas red area, PE Texas red height. And I'm going to make sure that we have our doublet exclusion. All right. Now, from there, Again, just a quick reminder, if I brought that gate out to include the aggregates, you see them in this plot right here. Okay, so we know and we have that confirmation that we're actually gating in the appropriate way. Now, some people, as they're starting off, would go ahead and take a look at their PE Texas Red and then look at that in a histogram and try and manually go ahead and set gates. But that becomes very hard to do at times, because where do we define our threshold for G1, G2, and the S phase? So as opposed to doing that, what we're going to do is we're going to walk through some modeling approaches that are available in FlowJo, which is the platform that we're using today. Okay. Now, before I do that, you can see I only have the gating applied to this one sample. So using the Shift key, I select all the gates and I'm applying that to all the samples. The quickest and easiest way for me to check my gates in FlowJo is to hit the Shift key and select this green arrow. And then all of my samples went, or all of my plots rather, I should say, went to the next sample. This gating is actually still pretty good. I don't need to make any minor modifications, with the exception of that right there. And if you do have to make any, that's okay. And then it becomes sample specific, okay? The only thing that really happens is that the gate is no longer bolded in your workspace. And if I wanted to kind of say, all right, you know what, on this one, I did not mean to change that scatter gate, you can delete it and it just snaps back to the original gate, okay? So from there, everything looks okay. Looks great. Now, we're gonna go ahead and look at the modeling to fit our cell cycle data, okay? I'm gonna close out of all of these plots because I no longer need them. We don't need to make our space any busier. And I'm gonna start off with the sample that I know is our gold standard, our best for this cell line. We see a nice distribution of populations. So that is the methanol fixed PI sample right here. I'm going to click on that last gate. And then from biology, in this tab right here, I'm going to go ahead and click cell cycle. Actually going to click it. Now what happens sometimes, bring this down because the Zoom window, is it's saying it can't calculate, unable to look at it based on what you've put into the, what your input was. What you see though is on the axis, there is nothing selected. So all you have to do is choose the appropriate detector. So I'm going to choose PE Texas Red by height. And what do we immediately see? We see a beautiful cell cycle profile, all right? What happens is it's modeling this data and it's fitting it to the curve. So what we're going to look at when we do this is a couple of different things. We're going to look at the RMSD, and that's the root mean square deviation. It's essentially telling us in very simple terms, how good is this data fitting, right? Now, if we had a poorer fit of our data and it didn't look as clean, that number is going to go up, okay? So the higher the RMSD is, the poorer the fitting of the data is. So typically I try and keep that in the single digits. It's not always going to be this clean, but that is something to be aware of. If you have an RMSD of like 30 or 40, probably not a great sign for the modeling. If you have an RMSD that's in single digits, that's usually a good sign, okay? Now, you also see here that there are different models. So there's the Watson pragmatic model, and that assumes a normal distribution. And it also assumes that you can identify one or two of the peaks, and it uses a Gaussian distribution for G01, and it sets the G2 at 1.75 times the G0, G1. You'd assume that that would be a perfect 2 times, but typically you don't see that it's exactly 2 times, and it's a little bit less than that, okay? The Dean-Jett-Fox model is slightly different. It's very similar to Watson, but from what I understand, it just fits the S phase of the profile a little bit differently. And there's some deeper mathematics we can dive into for that, but we're not going to for today. What I'd suggest is taking a look at both and see what gives you a bit of a better fit. So you can see here, the RMSD is 2.57 with the Watson, and the Dean Jet is a little bit higher at 2.71. And I'm going to proceed with the Watson, and I do see Derek, so I will.
Well, there's a couple of questions come through. And first one is about measuring a sub G1 population in FlowJo, and is there any option for that?
Sure. So you actually see right here, there's a percentage that's less than G1. So right now that's 2.84%, and you are able to see that. You know, you have to be careful when you're doing your scatter gating and going through all of your gating, because if you're very, very tight with your gating, you actually might be excluding some of those populations.
Exactly. And I think when I'm looking for sub G1 populations, I would gate slightly differently than I would if I was looking for DNA. You know, I would include most of the events that fall below the peak.
Exactly, especially
in
this
next, in that one. Yeah.
Yes, right there.
Yep.
Bring that down, and then you see that population changes.
Yeah.
So right now it's 2.84. For bring that up, you know, you can. Oh, was there a real change?
Yeah.
I guess not.
So again, it does depend on the question that you're asking.
Yeah.
And sometimes the further you get away from a classical G1 SG2, the harder the model has to fight to look at that. So there's another question there about deciding where the apoptotic cells are when you're modeling or when you're analyzing the cell cycle. If you're just looking at DNA analysis, that can be quite difficult. There's another couple of programs out there, there's a thing called ModFit and another one called Multicycle, which are really designed to analyze proliferation, whether it's by DNA content or by dye dilution, which have a lot more sophisticated algorithms built into them, which if I was looking for apoptosis, or I'm looking for aneuploidy or multiple populations, I would probably use those. So it depends which one I'm using. Actually, there's another question there. Why using height and not area for the modeling? In this case?
Right. Okay, that's a great question. So let's look. So if we do PE Texas red height, you see here, okay, and then PE Texas red area, they are similar, right? I tend to look at height when I'm looking at cell cycle data for nice tight peaks. Derek, maybe you want to chime in. I typically have used height. They should be somewhat comparable for this. If you have a specific reason that I might not be aware of, it's the
well, height,
I'll let you chime in.
Height is actually a better parameter once the object that you're measuring is smaller than the size of the laser beam. Okay, so if you're, for example, if you've produced nuclei here, height is probably a better measure than area. Area is better when the object is larger than the laser. Okay, so quite often with alcohol fixed cells, small cells, you could probably get away with either of those.
Yes.
Height or area.
Thank you. Okay. Any more questions before we keep going with the?
You can carry on, Kathy.
Awesome. So one other quick thing that I want to mention is that sometimes you might have a hard time pulling out and seeing such a clean modeling right away, right? So all I did was open it up and choose the detector that we wanted to look at, and that was it. Now, if I open up our, let me open up our M-phasor S right here, sorry, and go into biology and do cell cycle. Just bring that down. It's telling us that 94.9% is in G1. Now, what we want to make sure that we always do is run the controls. We've talked about that quite a bit today. But what good are our controls if we can't use them for our modeling? So in applications like this and experiments like this, what you can do in this software and in this program is you can go ahead and use this interval gate and actually put a gate around where the G1 range is. Okay, so by doing that, I'm saying to the software, G1 is specifically right here in this area and has a mean of 51,000, right? This is the population that I'm identifying as G1. Now, if I go ahead and I take this cell cycle profile that we've identified, and I take those settings with that G1 constraint and I apply it to all the samples, when I go back in and I look at that arrest, What is it now telling me? It's now telling me, because it has the appropriate reference for G1, that our G2 peak is at 93.3%. And that's a more accurate reflection, or it's a correct reflection of what we're seeing with this treatment that we've applied to these cells, where the mitotic spindles can't form, and we're seeing this arrest in the G2M phase. So there's a lot of cool things that you can do with this software in order to be able to pull out your populations and fit your data, okay? Now, there's a lot of other stuff we can get into, right? So you can set where you want your G2 peak to be. So for example, I said with the Watson modeling, it sets G2 at 1.75 of G1. If you ever wanted to modify and put those values in yourself, you could set it to a G1 by N and modify that. So 1.7, 1.9. You can go in and start putting in numbers to try and get a better fitting of the data for your specific samples. So feel free to explore these different applications. You could also put in what you believe to be the appropriate CVs. This gets into a little bit more of the technicalities of it. But again, if you see the RMSD is very, very low, it's in single digits, you're typically good. And you might be able to make some minor modifications to this, these constraints in order to be able to optimize the fitting a bit more, but these are available to you. What you can also do is you can copy the gates in the workspace. So you can see the frequencies and get that population percentage right there in the workspace. You can also open up your table editor, and you could just drag in the cell cycle analysis, and it's going to give you all the stats that are associated, the RMSD, G1 percentage, S phase G2, the means, so on and so forth. Okay. Before we wrap up though, I just wanted to show you one other cool thing that I thought would be very applicable for our session today. And this is opening up the layout editor. So we could look at a few overlays. The first overlay that we're going to look at is our methanol fixed sample. So if I go ahead and I drag this single cell PI population in, what we see straight away is that it's showing us all of the populations that are expressed, or sorry, the populations as they're distributed, I should say. If I double click this, I can take out some of that data. So I can uncheck show frequencies, population names, and gates, and annotation. And it's a little bit cleaner, right? Then hit OK. And then what I'm going to do is I'm actually going to bring in our M phase arrest sample, and I'm going to overlay that. And you can get a really nice visualization of how well that treatment worked in arresting those cells, right? So not only do we get our modeling, but we can also get a nice visualization of the data as well. What we can also look at for a really nice example of why we prefer alcohol fixation as opposed to our formaldehyde is looking at an overlay of the PFA compared to the methanol. So I'm going to drag in the single cells PI, make those same modifications, and then I'm going to drag in the PFA that was solubilized, and then it was formaldehyde fixed. And what we can see is we can see the difference in the same cells and what it looks like when you change how you fix your cells. Okay, so that was a bit of a quick overview, but now we've gone over how to go ahead and do your data analysis for DNA analysis by flow. Were there any other questions that came in?
Well, there was one that I sort of answered in chat about how can you differentiate cells that have doubled their genome, so cells that become tetraploid, to cells that are in G2. And we can't really do that just by using a DNA dye. We would have to add something else in there as well. But the DNA analysis can lead you down that path as to what to do.
Yes, absolutely.
Also, in cancer, what we can sometimes see is aneuploid populations, so cells that have not the normal diploid or tetraploid amount of DNA. But we do have some limitations with flow cytometry. We can't pick up single translocations, for example. It's not going to change the DNA content, but it is a very useful technique. I say I've been doing this for nearly 40 years, so it's kept me going.
I love you. Have a little bit more experience than me. That's why I defer to you for a lot of questions.
Yeah.
I know you're the
I
pro.
thought you weren't even born then, Kathy, and I started.
Close. All right.
Okay, so we've just got a couple of final slides because I'm aware we're approaching our 90 minutes. So
Stop my share.
I'll share. Oops, I'll share my screen. Okay, so it's just a summary, and actually this summary could apply to any experiment. Okay, it's not just a DNA analysis experiment. Remember, we always need to be aware of our cytometer, our instrument, which lasers, which filters we have in there, because that will, aligning that to what we know about our emission and excitation of our fluorochromes will tell us whether we can use those two things together. Of course, we always want to make sure that our cytometer is QC'd and performing optimally as well. With our cells, DNA analysis is no different to anything else. We need a single cell suspension. We've got to think about titrating and having the right protocols. And it can take some time sometimes to get it right, depending on the cell type that you're using. And although we've looked at propidium iodide here today, but we didn't have time to show you with some of the live cell DNA assays. That protocol I showed you for PI, that will work for any mammalian cell. Whereas if you're staining your cells with a viable dye like Herxt, you have to think about the concentration of the dye, the time of incubation, even the temperature of incubation. It's not quite as easy. And then as we saw with Kathy's section there, we have to make sure that we acquire our samples correctly, making sure we run them at the right flow rate, thinking about the number of cells we acquire. And then, of course, as always, we have to gate our cells. We have to cone down to what we're really interested in. So if there are any outstanding questions, and I don't think there are, a few thank yous coming in there, which is great. But if you do think of something later on, you can get in touch with us, and I'll give you the contact details in a second. But just to say that we will be making a recording of this available. It'll be on the OpenFlow Cytometry YouTube site. If you're not already subscribed to that, please. So everybody says, isn't it, at the end of the talk, please subscribe to my channel. We're not making any profit out of this, but if you subscribe, of course, you will get a notification when we upload this video, which will probably be towards the middle of next week. And we're not stopping here. We've got a next session on the 4th of March. We've planned that. And hopefully it's going to be an introduction to high dimensional analysis. We've done something really simple today, just one color. Some of the stuff we've done in previous webinars, we've looked at 2 or 3 or 4 colors. Many people are now doing a lot more than that. So a big issue in cytometry is how we analyze that. So we're going to start with a gentle introduction, hopefully, into the things that we need to consider in high dimensional analysis. And that very small URL there, registration is already open for this. But if you go to the What's On page at the Crick website, you'll be able to do that. But we'll be putting these things out in the usual social media channels. So if you don't already follow us on Twitter, please do so. You can contact any of us individually, or of course you can also email the openflow@gmail.com address, or comment on the videos. That's a useful way actually, because we will reply and that information is then available for everybody. So thank you all for coming. As usual, we've slightly run over time, not too bad today, but thank you all for coming. Hopefully that has been enjoyable and informative for you. And if you've digested all that and think of some questions, say, please do get in touch with us. So hopefully we'll see you at a webinar very soon.
Yes.
Take care out there.
Thank you, everybody. And thanks again to Quan Chung and the Ian Cheeseman Lab here for the samples. We really appreciate it and hope everyone's staying safe.
Absolutely. We'll see you all soon. Thanks, everybody, for coming.